Hi, my name is Leah Goodman, and I'm doing my biology role model project on Carol Greider. Greider was born in 1961 to two scientists. Her mother died when she was young, so she was raised by her father. She struggled in school, especially elementary school, because she had dyslexia. This caused her to have trouble sounding out new words. Instead, Greider learned to memorize words and letter patterns. In high school, Greider realized that her memorization skills were very useful in biology, and she fell in love with science. She went to UC Berkeley to study marine biology, and eventually switched to biology after working in labs under her late mother's colleague. She learned that she really enjoyed working in labs because there was a focus on interactions between people as well as work with science. Greider then earned her PhD in molecular biology at UC Berkeley, after having a hard time getting into graduate schools due to her low test scores. She worked in Elizabeth Blackburn's lab and aided in the discovery of telomeres, which earned her, Blackburn, and Jack Szostak the Nobel Prize in 2009. After graduate school, Greider conducted a fellowship at Cold Spring Harbor Laboratory, where she continued to work on researching telomeres and the role that shortened telomeres play in disease. Outside of work, Greider enjoys physical activity, especially swimming and spin classes. She also likes spending time with her two kids, and even took them to Sweden with her to accept her Nobel Prize. She is also an activist for women and minorities in science, and for using science to inform the public on issues. I admire Greider because she encourages both women and minorities to engage in science, and because she found a creative way to be successful with her disability. Greider currently works in the field of molecular biology, where she focuses on telomeres, especially how tumors use telomerase to continue their growth, and how we can use this knowledge to treat disease. Greider's most recent publication is an article with the title, TPP1 Promoter Mutations Cooperate with TERT Promoter Mutations to Lengthen Telomeres in Melanoma. TERT is a gene that produces telomerase, and mutations of this gene are often found in tumors. In this paper, it was shown that the expression of both TERT and TPP1 lead to longer-living melanoma cells. Figure A is a Western blot that shows that an increased expression of TPP1 leads to increased telomere length. Figure B is a graph showing that cells with both TERT and TPP1 expression had a larger population after 100 days than cells that expressed just one of those genes, or neither of them. Figure C is a Western blot that shows the expression of TPP1, TERT, both or neither in sample cells. The result on telomere length based on these different expressions is shown in the Western blot in Figure D. Currently, Greider's lab is focused on studying telomere length equilibrium, and using this knowledge to identify treatments for diseases caused by incorrect telomere length. Her current project is focused on, specifically, the relationship between DNA replication and telomere elongation.